Can you hear me well? Yeah. So thank for the organizer to bring up this nice line of presentations. So I'm going to present you some work that was done in collaboration with Munir and Mendali, Aude-Marie Grapperon, Wafa Zaraoui, Annie Verschuren, supervising Salma Aljan Haidia. And so we are from Marseille, from INT, and they are from CRMBM and the APHM. And so what we are going to, the question we are going to address today is how to efficiently extract some information from different MRI modalities, right? In order to characterize the stage of ALS at the individual patient level. And as we've seen in the different presentation today, so ALS has a lot of heterogeneity against different patients. And one problem, especially for neuroimaging, is we've got a small group size. As many of you may know, the conventional MRI is not used in the diagnostic, so it's only used to exclude some other potential pathologies. And what we are using is quantitative MRI that I'm going to introduce later to try to get some different views on the state of the brain. So what I'm going to present to you are preliminary results from this ongoing study. And as in the two previous presentations, we are going to look at classification for prediction. So I'm going to present two types of classification, patients versus control, and also stratification of the patient between slow and fast progression. And so, meanwhile, our techniques can also identify informative brain regions that are informative for the different classifications. So our cohort is made of 15 patients, and we've got heterogeneity in the onset site, in the disease duration, and in the ALS-FRS scores, revised, and the progression for these patients. So we separated for the progression the patient between slow and fast just by the median of this progression slope. And so we also have 14 controls. And the data were acquired in Marseille on a 70 scanner. And so we had three modalities of quantitative MRI. So one was fractional anisotropy, FA. The other one is quantitative T1. And both of them are aimed to capture the microstructure of the neuronal tissue. And another modality of MRI that we are considering is the sodium concentration that you can get with specific sequences. And this is a metabolic marker related to the sodium homeostasis and the mitochondrial functioning. All right. So these are some examples of images that we get from the scanner for this fractional anisotropy, quantitative T1, and the total sodium concentration. And for each of them, we actually have some 3D volumes there. And we separate the brain into different regions doing segregation of the brain, segmentation of the brain, in an atlas of about 180 regions that are both subcortical and cortical, and also involve the white matter. And so what we end up with for each patient is a number of values, one per region. And so we're collecting this data for all the different subjects, for the different regions. And so the point is going to be to predict for each subject a label, right? So here we've got our patients, our controls. And so we are going to train a mathematical function to use for each subject the different values that we have for the prediction. So if we are doing some conventional statistical analysis, we would look at, let's say, one region and look at the difference between the two groups, control and patients. Here we are doing predictions, so we are doing it in the other way. And so we could do that for all subjects as one, but instead, what we are going to use is a process in machine learning which is called cross-validation, where we use part of the subset to train the algorithm, and we leave a test set, which is in our case here just one subject out, to assess the performance. And by repeating this process for all the different subjects, then what we get is the test accuracy. And so this process is important when you want to understand how your predictive algorithm is going to generalize to new subjects. Okay, so that's not rocket science, but just the way to do to test the generalization capability. And so we can do that, can train our classifiers, different kinds of classifiers, so logistic regression or random forest here, on the different MRI modalities. So here what you have is the true labels in our data set, so patients and controls. And what you can see is that the two different classifiers, logistic regression and random forest, they capture different parts of the information for the prediction. They made errors in different ways. Also, when they are trained with the different MRI modalities, so TSC for the sodium, the quantitative T1 here, and the fractional anisotropy. And so the idea here is to try to combine the information here that we've got, especially in the different MRI modalities, to do the best prediction as possible. So to do that, we need to fuse the different information that we have. And so if you represent now the performance, so that means from each modality, we can count the number of good prediction, and we plot it here, so that are the red dots here. Yeah. And so to understand whether this prediction is good or not, what we do is we calculate a chance level, which is the distribution in gray here. And we do that by performing several repetitions of the shuffling of the labels that we train the algorithm with. And you can see that fractional anisotropy here is above chance, because the red dot is away from the gray distribution, as well as QT1, but for TSC, the performance is a bit lower. It's just outside the chance level. And now we can combine these three modalities to try to make the classification. And when we do that, we don't have the improvement of the performance, but we get further away from the chance level. So in a way, we have improved the statistical power of our prediction. Okay? So if I summarize for this classification of patients versus controls, so we've got some performance that is on par with previous studies. There were also some studies by Peter Bidi that had similar performance. So you can see that patients are well classified at 86% and controls at 80%. So you see that on each row, the error is on the other side. What we found is that then functional anisotropy was the most informative MRI modality. And as I said, we can also check which regions and which MRI modalities are informative for this. And we found that some gray subthalamic nuclei, like thalamus, part of the white matter, and for fractional anisotropy, and also some cortical region, precentral, frontal, and temporal, were informative for this prediction. And slightly negative result is that so far, we don't have a clear synergy between the different MRI modalities as we expected. So now let's switch for a more interesting classification, maybe, with the stratification of slow versus fast progression patients. And so we still consider the control here, and we are going to redo the same classification as before. And as you can see, now we've got 3 classes to predict, so the chance level is a bit below. And we can do that for functional anisotropy, quantitative T1, and total sodium concentration. And so when combining them, now we've got the same picture as before. We've got a performance that is at the same level of FA, which is the best performance that you've got across the 3 modalities. And we also have way above the chance level. So the red dot is away from the gray distribution. Now, one point that I find interesting is that if you don't consider the control but only the patient and try to separate them, this time you don't have statistical power because also you don't have so many subjects. So we can look at where this algorithm is making correct prediction and different and errors. And you can see that controls are well predicted here. And fast progressing patients are well predicted. And the problem mainly comes from the slow progressor. But if we think that from a clinical point of view, isolating the fast progressing patient is more interesting, then that's fine because we know that controls are controls and patients are patients. And then using this algorithm, we can still clearly divide with this line and isolate or identify the fast progressing patient. And as you can see, different modalities and different classifiers, again, give different views on these patients. And now our next point is to try to work better to make a fusion of these different algorithms and these different modalities to improve the diagnostic. So I've shown you some results that are promising to identify patients with fast progression. We've seen that patients with slow progression are closer to the control group, which I think is in line with the literature. And so now we want to change to improve the way we evaluate the progression scores to do some prognostic more in the future. I've highlighted to you the importance of taking the control in this prediction. And that also goes in line with the need to increase the data set to improve the statistical power. So that's underway with the collection of new samples. And so far, the MRI modalities that are more informative are those FA and QT1 that are related to the microstructure of the neuronal tissue. And in the future, we want to refine the brain parcellation, especially because for sodium concentration, that seems to be interesting. And if you want more detail on that, go to see the poster by Munir and Mendeli. And we want to also combine later with functional MRI in stratigraphy. And I would like to thank the sponsor, especially Arsla, who gave us some funding to pursue this work, and thank you for your attention.
Thank you very much.